Yeah, so I'm Heather. I'm a fourth-year graduate student in the genetics department over on the medical school campus, and I study what's called RNA splicing. I think it's this really, really cool phenomenon that's often overlooked when people are thinking about genetics, but I hope by the end of this talk you'll appreciate how important it is. Before we get into what splicing is, I'm going to pose a question. Who has more genes? We're comparing this nematode on the left that's only about 1 millimeter in length. It only has about 1,000 cells, and it's pretty boring. It just eats and sleeps and makes babies, and we're comparing them to us humans. We are almost 2 meters in height. We're made of trillions and trillions of cells, and we have lots of traits. We have different hair color and eye color and skin color, and we're really, really complex. So who has more genes, the nematode or the human? So I'll try and get an answer by raising hands. So if you think that the nematode has more genes than the human, raise your hand. All right, a couple. Who thinks that the nematode and the human have the same number of genes? All right, a few more. Who thinks that the human has more genes than the nematode? All right, this is actually pretty mixed results. So I think most people would answer that humans should have more genes than nematodes, because genes are what make our traits, and humans have so many more traits. We're complex. We do all of these different things. We're able to read and write. But in reality, nematodes and humans have about the same number of genes. And this really, really surprised scientists when we first discovered this, because we thought that genes were so critical for making humans humans, but in reality, other things such as RNA splicing are what allow humans to be so complex without having many, many genes. And we'll get into really the complexity of this in the second part of the talk tonight. Come in. Here's the roadmap for the evening. We're going to first discuss what is splicing, and then get into what is alternative splicing. And this part of alternative splicing is really going to tackle that question that I just addressed. And lastly, we'll discuss what happens when splicing goes wrong. How is splicing related to human disease? First, to address what is splicing. Here's a really simple cartoon of a cell. Our bodies are made up of trillions of these, and most of our cells have a nucleus and DNA inside the nucleus. And DNA here is represented by these squiggles. And you also often see it as this double helix if you zoom into the DNA structure, and that's because that's what it looks like. It's this really beautiful double helix molecule. As scientists, and especially as geneticists, we think of DNA actually as a string of letters that holds important information for making the proteins in our cells. So how does DNA make proteins? Well, along DNA are these particular regions, and those are called genes, and the genes delineate where the proteins are going to be made. These genes get copied into RNA, and then the RNA is read and made into proteins, and then the proteins perform function. In order to simplify this a little bit, I'm going to make an analogy to something that I like to do a lot, which is cooking. So cooking, if we want to make a meal, often we'll refer to a cookbook or a recipe book that will have all of this information that we need in order to cook a meal. But we're not going to cook every recipe in the book for dinner. That would just be way too much food. So we'll pick out one recipe that's in the cookbook, and that's like a gene. A gene is like one recipe within the cookbook that is DNA. But I'm a really, really sloppy cook, so I don't like to take my recipe book into the kitchen with me. Instead, I like to copy the recipes onto a piece of paper and take that into the kitchen, because then I can just throw it away after. So the copy of the recipe is like RNA. It looks a lot like the DNA, but it's just a copy of it. And then when you make proteins, that's going to look a little bit different. So the protein is actually the food that comes out. So the protein is resulting from the copy of the RNA. And the protein is what serves the function. And in this case, the function is making me full for food. So when I'm referring to these things of DNA, RNA, and protein, think about the analogies to cooking as to what purpose these things are serving. So what does DNA, RNA, and proteins actually look like? So before I referred to DNA as kind of a string of letters. So if we think of letters of DNA. This is just one example. There's 2 strings, one on top and one on bottom. We often refer to these as strands of DNA, and these strands are really important. There's always matching of the DNA. The DNA is made up of 4 different letters, A's, T's, G's, and C's. A's and T's always match up, and G's and C's always match up. So it's important that DNA has 2 strands. The RNA looks a lot like DNA, but it only has 1 strand. And it's essentially a copy, this is essentially a copy of that top strand of DNA, except for one difference. So one of the letters in RNA looks a little bit more different. The molecule looks a little bit more different, so as scientists refer to that letter a little bit differently. So now instead of T's in the RNA, we have U's. But RNA mostly looks really similar to DNA, it's just 1 strand. Proteins look a lot more different. For every 3 letters in RNA, we make 1 protein molecule, and protein molecules also have different letters, and these letters are represented in different colors here. The sequence of all of these things are really important. If one of the letters on DNA is changed, in this case a G is changed to an A, that will then get copied into the RNA, and then the protein sequence here will also be changed. And this can cause really big problems if that letter on the protein is really important, or it might not really have a big effect. In this case, perhaps it changes the protein in a slight way where it can't form its normal structure and it might be a little bit misshapen. So that could be the result of a change in the sequences here. Now if we add another change, we add a G here, that G gets copied into the RNA. Now this signal in the RNA is really problematic because it will tell the machinery that's making the protein to stop here and don't make any more protein. And that's really problematic because the cell sees that. The cell sees that the protein is too short and will completely get rid of the protein. So now that there's no protein resulting from this. So you can see how sequence is really important if you have these this information in the sequence that's telling the protein not to be made, then that can have really big problems if that protein is important. So now we know that sequence is really critical and all of these things are made up of different types of sequences. So let's dive into what these genes really are. So here's a DNA, and DNA, like I told you before, is made up of sequence. Parts of the sequence is really important for certain things. So part of the sequence will tell where a gene starts and where a gene stops. Other pieces on the DNA indicate really important regions that are important for making the protein. These are called exons. So all of these exons are important for making the protein. The pieces in between the exons are called introns, and these are not really important for making proteins. So we have our gene here, has a start position, a stop position, 3 exons, and 2 introns. Now if we're going to make a protein from this gene, first we need to make RNA. So the RNA starts being made at the start position, it stops being made at the stop position, and the 3 exons are copied and the 2 introns are copied. What happens next is really, really cool. So these intron pieces that are in between the exons get taken out. There are these molecular scissors that cut them and remove them, and they're totally taken away. And then the molecular glue bottle will paste the exons together. So now we get rid of the introns and are only left with exons, and these exons go on to make protein. So the protein is now only made up of the exon sequences, and the introns are totally gone. This process in the middle is called splicing. So this is what the whole talk is about. Removing pieces out of the RNA and putting other pieces together in order to form different RNAs that will make protein. A little bit more terminology now. So we have 2 different species of RNA here. We have this RNA on the bottom, which is only made up of these exons, and this is the more mature form of RNA, so we call it mRNA for mature RNA, and it's completely spliced. Before you get mature RNA, you get premature RNA. So this unspliced version is called pre-mRNA, or unspliced. And I will refer to these as the bottom one as mRNA, the spliced version, and the top version as pre-mRNA. So you can see this is pretty complicated. Why would we cut out pieces and put other pieces together? You might be thinking this can't be happening a lot throughout our bodies or throughout our genes. Actually, splicing is really, really common in humans. So here are different species, and for all of these species, I'm outlining in this bottom how the average number of introns for each of their genes. So bacteria, they're single-cell organisms. They don't have any splicing for their genes that go on to make proteins. Yeast, this is the common yeast strain that we use in the lab, but also what you would use to bake bread. On average, in their genes, they usually have less than 1 intron per gene, so they don't have very much splicing. The nematode that I showed you on the first slide, they have more splicing. So on average, for their genes, they have about 5 introns. But us humans have more. We have about 8 introns per gene. And if you think about us having 20,000 genes and 8 introns per gene, that's a lot of splicing that has to happen in order to make correct proteins. And some of our genes have almost 100 introns. So that's a ton of splicing that really has to happen. So you might be wondering, what's doing all of this? Or how does splicing really work? Well, I showed this simple diagram of molecular scissors and molecular glue bottle, but you're probably thinking that's not really how it works. Instead, splicing looks a little bit more like this, where it's this really large complex that globs around these exons and the intron and directs the intron, takes it out, and totally gets rid of it and fuses the exons together. It's a really, really complicated model, and scientists are still trying to figure out exactly how it works. But this whole complex here is called the spliceosome. I think it's a pretty cool name for splicing. The spliceosome is the multipart machine that directs splicing. It's made up of hundreds of proteins, so there's really a lot of proteins here. And there's really 5 core components that are made up of proteins in RNA that are really directing the active splicing reaction. But in total, some of these proteins are physically involved in splicing, and some of the hundreds of proteins are more indirectly responsible for regulating splicing. And you'll see why those proteins are important for regulating splicing when we talk in the next section about alternative splicing. To summarize this first section, we talked about how splicing is the cutting pieces of RNA and pasting other pieces together. That splicing is very common in the human genome, and that the spliceosome is the large machine that operates the splicing events. So now I want to take any questions that you have about the actual splicing process, if you have any. Yeah?
Did you say there's an average of 8 introns per human gene?
Yeah.
So is that 9 exons then?
On average, yeah.
Okay.
Yeah, so typically there's 1 more exon than introns because the introns are in the middle. Mm-hmm. Yeah?
Is this related with the transposons?
Yeah, so this question is it related, is splicing at all related with transposons? And quick answer is directly, not really. So the process of like cutting out transposons and putting those in, like moving those around DNA and splicing are pretty different. But there are theories about how the active splicing and through evolution splicing may have arisen through transposons. But quick answer is like the actual mechanisms are very different. Yes?
So the protein product is the result of the spliced RNA, and are the exons reordered, or are they just like spliced and like truncated?
So the question is, when you have this mRNA that's made of exons, are they always in the correct order, or are they ever reordered? In the case that I showed now, they're always in the correct order, and typically they always form in the correct order. What you might find really interesting in the next section is with alternative splicing, you'll see that the order doesn't always stay intact.
And you can make proteins in its custody by this process?
So this question is, can you make proteins in a test tube by this process? So the really interesting thing about splicing is that it's so complicated that scientists have not been able to fully recapitulate splicing just by adding the components into the test tube. What you have to do is actually break open a cell, add everything that the cell has in order to make that reaction work. So hopefully there will be a day soon that we really understand every component that's absolutely necessary for the splicing reaction for us to be able to do it in a test tube with everything we know, but that has yet to happen.
So it's a living reaction.
Yes.
So that's a lot.
It's one way to think about it, yeah. Yeah.
Does splicing happen inside or outside the nucleus?
Great question. That's close to what I work on too, so I like that question. The question is, does splicing happen inside or outside the nucleus? And I guess to make a simple answer, I think we don't totally know. Most of it definitely happens in the nucleus, and a lot of it is thought to happen while the RNA is even being made. So I'd say that most of it happens at that point. It could be that some of the splicing does happen outside, and it's thought that some of it does happen outside the nucleus as well. Yeah.
So splicing always leads to full form of protein, or you will get like truncated protein, or?
Great question. So the question is, does splicing always lead to the full form of the protein? And the answer is, stay tuned for the rest of the talk, because we'll see how it might not. Yeah. All right, so we'll stop the questions there and move on to the next section, and can bring up other questions later on throughout the talk. So the next question is, what is alternative splicing? Alternative splicing occurs when multiple distinct mRNAs are made from the same gene by splicing different combinations of its exons and introns. So what does this look like? We have the same gene that we had before, where we have the start and the stop and the 3 exons. We make our pre-mRNA from this gene, and this pre-mRNA looks like it did before. Now when we splice the RNA, we're going to splice out those introns. So we have the scissors cutting out this intron, cutting out this intron, leading to an mRNA that has the 3 different exons. But this might not be the only way that we have to splice this pre-mRNA. What if we move this scissor to the outside of that yellow exon? Now we're cutting out this intron and cutting out this intron along with this yellow exon, leaving an mRNA that only has the orange exon and the blue exon. Lastly, we can cut in a completely different way, where now we are only cutting to the right of this orange exon and to the left of the yellow exon, cutting out this entire sequence in the middle that contains both introns and the blue exon. So now we have an mRNA that's only the orange and the yellow exons. So you can see here that we have multiple different mRNAs, all coming from the same gene. And importantly, these 3 mRNAs make 3 completely distinct proteins. And you can imagine how these proteins can go on to serve different functions. So in this way, we go from 1 gene to 3 proteins, and this is all through alternative splicing. For another piece of terminology, these mRNAs, I'm going to refer to them as isoforms. So basically, they're just different forms of the RNA coming from the same gene. So this is one simple example of alternative splicing on a short gene. But what's kind of the potential for alternative splicing? Here is a pre-mRNA that's actually from a gene in fruit fly called DSCAM. And the gray bars here represents exons that are not alternatively spliced. The colored boxes or rectangles represent exon regions that do have alternative splicing. Here are these numbers represent the number of alternative splicing options for each of the different regions. So this orange region has 12 different alternative splicing options. Blue region has 48 different alternative splicing options. Green region has 33, and the purple region has 2. So these all have a fair amount of alternative splicing options independently, but if we want to calculate the amount of alternative splicing that happens for this one gene, we would have to multiply them all together. And this gives you 38,016 different alternative splicing isoforms. So that's a ton of information all coming from one gene. To put this in some perspective, the fruit fly genome, the number of genes that they have in their genome is only about 17,000. So we're getting from one gene twice as many isoforms that go on to make proteins. So that's a ton of information that's just all coming from splicing and not necessarily just coming from the gene information. So if we can make all of these different isoforms, can they actually serve different functions? Here's an example of how an isoform, or different isoforms coming from the same gene serve different functions. So in our bodies, we have different types of muscle. We have cardiac muscle in our heart, smooth muscle in internal organs, and our skeletal muscles. And in each of these muscles, we have cells that need to perform important contractions. And those cells need proteins to do that. So there's one gene, tropomyosin, that is made into protein in each of these different muscle cells. And it's alternatively spliced such that it makes a completely different protein in the 3 different muscle cells and serves a different purpose for the contractions of the individual muscles. So here's a case where you have one gene making different alternative splicing isoforms that go on to make proteins that are really important and serving different roles. So we are not the only ones that think alternative splicing is really powerful and want to use it. Viruses also take advantage of alternative splicing. So viruses, when they infect a host cell, like a human cell, they will make DNA that will get incorporated into our own genomes. This DNA gets made into RNA, and then the RNA then the virus RNA uses our own splicing machinery, or our own spliceosome, to splice the RNA, and this will result in different proteins and RNAs that are really important for the virus to continue to propagate. So this is really advantageous for the virus because instead of carrying a ton of information, genetic information to start, they can carry less genetic information and splice it in different combinations such that it still makes the necessary proteins and molecules that it really needs to move on. So how common is alternative splicing? I showed you before in bacteria and yeast that they don't have very much splicing. They have few introns per gene, or 0. So they don't really experience alternative splicing. Now we're going to compare the nematode that I showed you on the first slide with the human. It's estimated, and this number can change as alternative splicing is studied more in nematodes, but it's estimated that up to 25% of nematode genes undergo alternative splicing. And they have fewer introns per gene, so there's less potential for alternative splicing. Humans, with our on average 8 introns per gene, we have up to 95% of our genes undergo alternative splicing. And this is kind of crazy, but if you think about, we have trillions of cells. For all of these trillions of cells, we might need completely distinct proteins in the cells. So it's actually not too surprising that most of our genes undergo alternative splicing so that we can have all of this complexity in all of these different proteins. So what does this mean? Like, how many proteins do you think humans actually make? So if we have one gene here, and that can go on to make anywhere between around 1 to 1,000 different mRNAs. But we don't just have one gene, we have 20,000. So if we consider our 20,000 genes and how many RNAs might be made, approximately 250,000 to 1 million different RNAs or mRNAs are made throughout the cells in our body. And this is representative of anywhere between 250,000 to 1 million different proteins. So I showed you before a slide where we were comparing the nematode to humans and showed you that we actually have the same number of genes, but in reality it's likely that humans have way more proteins than the nematode, and this is all due to alternative splicing. So you can see that alternative splicing is really, really powerful for creating all of the protein diversity that we have in our cells. summarize this section about alternative splicing, we learned what alternative splicing is, that it occurs when one gene makes multiple different mRNA isoforms through splicing different forms of RNA. That mRNA isoforms from the same gene can make multiple distinct proteins that have distinct functions, and that alternative splicing allows for many more proteins to be made than we have genes. So now that we've learned about alternative splicing, are there any questions on this topic? Yeah?
When you had the slide with 4 different exons,
Mm-hmm.
you had different numbers associated with how many, I guess, splicing options there were. How did different exons have different splicing, number of splicing options?
So the question is, how do different exons have different splicing options? So that was an interesting example where those boxes represented more regions of exons. So one region really has in the case of the 12 different options, it would have 12 different exons, and one of them would be made for each of the different cases. So it was just a simple way of representing it. But to kind of answer your question further, alternative splicing, I presented it kind of in a simple view, but there are a lot of ways of alternative splicing. So you can just completely skip exons, or in some cases you can kind of cut an exon in half and only have half of it be made into the protein. So really there's a lot of options when it comes to alternative splicing, and it's not always just the simple idea of oh, let's just skip this one exon this time, or keep this one this time. Yeah. Yeah?
If you have a mutation on an intron, would it never matter?
Good question. So if you have a mutation on an intron, would it never matter? In the next section, we'll talk a little bit about that, because there are some parts of the sequence in the intron that are important for splicing, even though they're not important for making the protein. I think this to answer this in a more broad way, our human genome is really made up of mostly non-protein coding sequence. So only about 2% of the whole genome goes on to make proteins. But a lot of the other parts, like the intron sequences, are really critical for other aspects of biology. So there can be things within the introns that might not be critical for making the protein, but are critical for other aspects of other aspects of cell biology. Yeah.
Are there ways to control the alternative splicing? Make some things more preferred than others?
Good question. So the question is, are there ways to control alternative splicing? There definitely are, and it's really complicated that I didn't want to get into it too much in this talk. So there are certain sequences along the RNA that will guide the spliceosome into splicing certain ways, and that's kind of how. And then also, if you think about different cells, how to make different isoforms in different cells, if you have certain proteins in one cell but not in the other, that can also dictate how a splicing reaction will occur. And then yeah, there's really so much that is going into it, and so much that people are still studying. But I think one aspect that might interest you is in the next part of the talk, I'm going to talk about kind of a therapy that's trying to trick splicing into making different RNA products.
Splicing depends on the length of the gene. Is there any correlation?
Good question. Does splicing depend on the length of the gene? Not so much the gene length, but there are now studies looking at intron length and whether that affects splicing. A cool finding especially in Drosophila, and it happens in humans, I believe, too, that there are some introns that are so long they can't be spliced all at once. So they'll kind of be spliced in different regions, and so the first part of the intron will be taken out first, then the second part, and it's called recursive splicing. So there are mechanisms for dealing with really, really long introns and things like that. All right, so now it's time to take a short intermission, 5-10 minutes. Before the break, we discussed how splicing is very important. On average, we have about 8 introns per gene. most of these genes undergo what we call alternative splicing, and due to alternative splicing, humans have many more proteins than we do genes. Because splicing is so important, so abundant, and so complex, you can imagine if there are mistakes in splicing, it could have really severe consequences. Here's a list of all of the genes that are in some way impacted by splicing. So mistakes in splicing either cause these diseases or affect the prognosis in some way. So splicing is very, very relevant when we think about disease. Mistakes in splicing can occur in two different ways, or two main ways. The first way is when there is incorrect splicing of a gene. So if one gene doesn't splice properly, that won't make the correct protein, and that can be problematic if that protein is important. Splicing mistakes can also occur if the spliceosome or any part of the spliceosome is impaired. And this would cause impacts for more genes, as the spliceosome is important for splicing many genes. First, we're going to talk about the first part, when there's incorrect splicing of a single gene. Case study to describe this, this concept is with muscular dystrophy. Muscular dystrophy is the slow weakening of a muscle where it starts from a really strong muscle and starts to get weaker and shrink into a less strong muscle. There are two main versions of muscular dystrophy disease that I'm going to discuss today. First is Duchenne muscular dystrophy, or DMD. This affects 1 in every 5,000 males, and it leads to very severe muscle weakness. The age of onset for Duchenne muscular dystrophy is about 4 to 5 years old, and most of these patients have trouble walking by the time they're 10. So it's really, really severe and a sad disorder. A different type of muscular dystrophy called Becker muscular dystrophy, or BMD, affects 1 in every 50,000 males, so it's more rare. It results in a milder form of muscle weakness, and the age of onset now is about 10 to 20 years. So this is a less severe and more comfortable form of the muscular dystrophy disorder as compared to Duchenne. The important thing to know is that even though these are different disease types and different prognosis, they're actually caused by mutations in the same gene. The gene that's affected by in these patients is called dystrophin, and it's one of the longest known human genes. And this is just a really simple schematic up here. The blue boxes and rectangles represent exons, but because the gene is so large, I couldn't fit it onto one slide. So this is just a really simple schematic that's not to scale. In dystrophin gene, there's 79 exons, so you can imagine a lot of splicing has to happen correctly in order for it to be the right form. And I'm not going to totally get into what the dystrophin protein does, but it's important to know that it stabilizes muscle fiber to the cell membrane. So if it's not there, then the membrane will start to break down and the cells will die. So if you don't have the dystrophin protein, or if it's not correct, it'll lead to cell death, and that's why you get shrinking of the muscle. So how do mutations in the dystrophin gene lead to muscular dystrophy? If we have the dystrophin gene and it's completely normal, there's no mutations, it will lead to the normal dystrophin protein, and these individuals will be very healthy. If there's a small mutation in the gene and you still get protein, but it's a little bit different or a little bit misshapen, this is what leads to Becker MD. So the dystrophin protein is still there. It can still have partial function, not full function. So it still leads to muscular dystrophy, but it's not as severe as Duchenne muscular dystrophy. Now, if there's absolutely no dystrophin protein from this gene, then this is what leads to Duchenne muscular dystrophy. So if you don't have the dystrophin protein, then it will lead to rapid breakdown of the cell membranes. Cells will start dying and the muscle will shrink much more rapidly in the case that there's no dystrophin versus where there's misshapen dystrophin. So how do mutations in the dystrophin gene actually cause this? So now we're going to look at splicing defects and how they can be caused in the in the dystrophin gene. So if here's pre-mRNA and the exons are the blue boxes, if we zoom into one portion here, we're looking at one intron. And if you remember from this first section, we talked about how sequence is really important in RNA and how the RNA is made up of the sequence of letters. So now here I'm showing some letters within this intron, and these are the letters that are really, really important for splicing. So we really need these letters here for splicing to work properly. So if you have all of these letters in the right places, it leads to correct splicing. Now if we change a letter, like in this case we change the A to a C, it leads to incorrect splicing. Also, if we change this G at the end to a T, it also leads to incorrect splicing. So now you can see how the sequence in the DNA isn't just important for making the sequence of the protein, but it's also really important for splicing correctly. So now if we have a mutation, one of those sequences, really important sequences here, this leads to an mRNA that might keep that intron there, which it was originally supposed to splice, and this can be really problematic. If it's a short intron, it might lead to just a misshapen dystrophin and Becker MD, but in most cases, if this isn't spliced, then it would lead to a no dystrophin being resulted from this RNA, because there are signals here that says that this is really wrong and the protein is probably wrong, so it won't result in any functional protein here. So for muscular dystrophy, not all mutations are in splice sites. This is just one example. And what I want you to get from this is that mutations in splice sites can be more problematic, because instead of just changing one letter that changes one letter in the protein, now that one letter changed causes a large insertion or a large removal of a whole sequence of RNA, and that can drastically change the protein. So it's really, really important that splicing happens correctly in order to make correct protein, because otherwise it can lead to diseases. But before leaving on that sad note, I want to tell you a really cool story about how scientists have developed a way to potentially trick the splicing machinery in order to make more correct versions of protein. So here is a version of the mRNA. Now we have a different mutation. This mutation enters a stop signal in the RNA, and if you remember from the first part of the talk, if you have a stop signal, this often results in no protein being made. If we zoom into the stop signal, it's in this exon. So ideally, we do not want this stop signal to be made into the mRNA, because if it's there, it will signal the protein production to stop, won't make any more protein, and typically that protein will just be abolished and not result in any functional form of the protein. So ideally, when we're making the mRNA, we want to just remove this exon and get it out of there, because it's going to be more problematic with it there than with it not there. So what scientists have developed is this tool called antisense oligonucleotide, or ASO. What this ASO is it's really just a synthetic sequence of DNA that can bind to the pre-mRNA. So if you remember from the beginning, pre-mRNA is one strand. So it's one strand of sequence in this here, the top strand, and that can match, but it can pair with a strand of RNA or DNA. In this case, it's pairing with a strand of DNA. And this binding is actually really strong. So scientists developed this antisense oligonucleotide right here because it's masking a signal that's in the pre-mRNA, and that signal is telling the splicing machinery, or the spliceosome, to keep this exon here. But if you mask that signal, then the spliceosome won't recognize that, and it will skip this exon. So what this looks like is, if you don't have the ASO, the signal will still be present, and the spliceosome will cut out this intron and cut out this intron. But if you mask that signal with the antisense oligonucleotide, now you don't have cutting here or cutting here, so you're just cutting out this big region and totally skipping over this exon that has the stop signal in it, which is really problematic. So to look at this again, if we have a pre-mRNA and the orange exon is the one that we don't want, if we don't add the antisense oligonucleotide, that orange exon will be maintained in the mRNA, and this will result in no dystrophin protein being made. And these patients have Duchenne muscular dystrophy. Now if we add the antisense oligonucleotide, it completely skips over that orange exon. And now we do get protein. It might be a little bit misshapen because we're missing that exon, but we do get protein. So with this antisense oligonucleotide, you can convert patients that have no dystrophin and exhibit signs of Duchenne muscular dystrophy into patients that have some dystrophin and might, even though it's a little bit misshapen, they have a less severe form of muscular dystrophy. This is a really cool concept that actually came to fruition this fall. The FDA granted an accelerated approval to the first drug, which was an antisense oligonucleotide for Duchenne muscular dystrophy. This drug is called Exondys 51 because it does pretty much what I just told you. It skips an exon in dystrophin, so exon is exon and dys is dystrophin, skips exon 51 in dystrophin. And it was designed by Sarepta Therapeutics here in Cambridge. This story was a little bit controversial because there weren't very many patients in the trial, and some of the science is a little bit controversial, but because there's currently no other drug for Duchenne muscular dystrophy and there weren't horrible side effects, the FDA granted this with an accelerated approval and is still going to constantly be monitoring it over the next 5 years to make sure that it's still doing what is expected. But the nice thing is that it does show promise, and it does show promise for other diseases, how you can kind of trick the splicing machinery into making better versions of mRNA and better versions of protein that they weren't making on its own. Does anyone have questions about that story about splicing defects for a single gene, or muscular dystrophy, or the Exondys 51 ASO? Yeah.
How does that get delivered to the patient?
Yeah, so I believe it's so the question is, how does this get delivered to the patient? And I believe that it's either through injection or IV. That's the most critical part of any type of oligo type drug, is that the cell has a lot of systems your whole body has a lot of systems to not let DNA or RNA in, because those are often in the form of viruses or other things that you don't want. So there's a lot of degradation can happen. So that's going to be the next, that whole concept of making sure that delivery happens well is a really important part that needs to be worked out soon. Yeah.
So do you have to inject locally in those muscles?
I believe the way that some of the trials were set up was through injection into muscle. And then they would test that muscle to see if it started to produce dystrophin. But I know that was one trial, and I think another trial did a more systemic version, but I'm not totally sure on the details. Yeah.
Does there have to be like a schedule or multiple injections over a period of time, or is it just one time?
This is multiple, I believe. Yeah. Because it's not producing it. If in some way you can develop a system where you're constantly producing that ASO, then you wouldn't have to do it multiple times. But in this case, you're not producing it, so it runs out. And if you need to make the dystrophin protein, you need that there.
So it'll be indefinitely over the course of a patient's life?
Yeah, I believe so. Yeah.
What was the term you used for that class of drug? You said it was like oligo something type.
Antisense oligonucleotide. Yeah.
But you described like that type of drug generically. It sounded like we were talking about in terms of those types of drugs.
So like any type of DNA or RNA based therapeutic, that's probably how I referred to it as. So antisense oligonucleotide, basically what it means is it's like DNA or RNA, and it's in the orientation such that it can bind to RNA. Yeah. Any more questions? Yeah.
So the reason that they have to be injected, is it because the human DNA want to go back to the default? Is that why they have to continue injecting it?
So the question is, basically the question is, why do we have to keep re-injecting it? So you have dystrophin protein in your cells, but you constantly have to be making it in order for it to be there because it will break down. And so if you have this ASO, that will also eventually break down. So we need to re-inject it or re-add it so that you can continually be making RNA and splicing the RNA correctly and making the dystrophin protein, because your cell is not actually making that antisense oligonucleotide. You have to add that exogenously.
So what is the interval? Is
Ooh.
it the same for all the medication like that, or is it different?
So the question is, what is the interval? I don't know exactly the specifics of it. I'm not a doctor. I don't know exactly how these patients, how to deal with these patients. But I think a lot of those types of questions are still being worked out because it's still going through trial. So the actual doses, how many times to treat them, those are all really important questions that actually need to be worked out through many tests and many trials. Yeah.
I understand you said that this research was not done very thoroughly, but in general, the side effect, is it related to muscles, or it could be completely unrelated to that specific issue?
The question is about side effects or it not being very thoroughly. So the issue with this case and many other rare disease cases is that, so this is for muscular dystrophy where there aren't very many patients, so there's or not very many patients that are susceptible to this exact drug because it's skipping one exon. So the dystrophin protein is really long, or the gene is really long, so not every patient has the same exact mutation in the dystrophin gene. So because not every patient has the same mutation, this antisense oligonucleotide might not treat every single patient. In this case, it only treats about 13% of all patients with Duchenne muscular dystrophy. So because it's not treating every patient, they didn't have very many patients for the trial, and that's more the reason why the results were not as conclusive, because if you don't have large numbers of patients, then you don't.
I'm saying the side effect, is it usually muscle related because it was for muscle, or it could be like related to, I don't know, legs, or like, I don't know, skin, or heart, or something like completely different?
It could be related to other things. In this case, they didn't notice very many side effects. Yeah. That's part of the reason why it was approved. Yeah.
Was only that 13% of the patients who have that specific mutation being treated? So they were using like a diagnostic or something to make a diagnostic to figure it out?
Yeah, so the question is, how do they pick the patients that are treated? So this antisense oligonucleotide was designed to skip exon 51. So what they did is look at the patients to figure out where their mutation was, and if it was in that exon, then they're part of that classification. And it just happened to be that they picked the exon to skip where most patients have a mutation, which makes sense because you want to treat the most patients possible.
So 13% is the most, is the largest percentage, or the largest group?
Yeah.
So you could make a similar therapy for the other ones?
Yeah, so in theory, because it's just based on sequence, it should be straightforward to adapt this technology to another exon, just based on the sequence. But a lot of testing has to happen before you go from that into a patient. All right, let's move on to the next story, and we can ask some more questions after if you still have them. All right, so now we're going to talk about what happens if the spliceosome is impaired. So if we remember, the spliceosome is that really large multipart machine that really directs splicing events. So we can imagine that if any component in the spliceosome is not working properly, then it would lead to incorrect splicing. And this can be really problematic because it might not just affect one gene, but it can affect many genes because splicing is important for many genes. What was found recently is that splicing factors are frequently mutated in some cancers. The cancer that I'm going to talk about now is myelodysplastic syndromes, or MDS. This is a cancer that starts in the bone marrow where blood cells are made. There are about 13,000 new cases of MDS in the United States each year. Most of these cases occur in patients that are 65 or older. A third of these MDS cases go on to progress acute myeloid leukemia, which is a more severe blood cancer. The important thing in relation to splicing is that 50% of patients with MDS appear to have mutations in key splicing factors. So the obvious next step is, what are these mutations and what are they doing, and are they actually the cause of cancer? So a really nice study 2 years ago in 2015 looked at splicing factors that are mutated in cancer and recapitulated this cancer in mice. So they have normal mice that have the normal intact spliceosome, and they also have mouse where they add a splicing factor that is mutated. So now we have a mouse with a normal spliceosome and a mouse with the same mutation that's in the cancer of humans, in a mouse with a splicing factor mutation. In their normal mouse, they see normal blood production in the bone marrow, as expected. Now, in the mouse with the splicing factor mutation, they see abnormal blood production and MDS-like features in the bone marrow. And so here, with the same splicing factor mutation in mouse, they see a recapitulation of the same signs of what's found in the cancer patients in humans with these same splicing factor mutations. So the next question is, what is the splicing factor mutation actually doing? Is it really affecting splicing? So again, if we have the normal mouse and the mouse with the splicing factor mutation, now if we look at their RNA, their pre-mRNA looks the same. When you look at spliced RNA, there are differences. So the normal mouse now has skipped this blue exon, whereas the mouse with the splicing factor mutation keeps that blue exon. And they found this across many, many genes in these mice, where the normal mouse tended to skip exons and the mouse with the splicing factor mutation tended to keep the exons. So now the question is, well, how is this actually related to cancer? Is just the fact that there's the splicing is different, is that what's causing it? Or is there one gene where splicing is different, and is that what's really causing the cancer? So what they did is they found a gene that they think is probably causing the cancer. And what this gene looks like is, if you have the normal mouse and the mouse with a splicing factor mutation, in the mRNA, the normal mouse has skipped a blue exon, and the mouse with a splicing factor mutation keeps that blue exon. But that blue exon is really problematic because it has a stop sign in it, and that means that no protein is being made. And so now if there's no protein of this gene for the one with the splicing factor mutation, but protein for the one with the normal mouse, so then they looked at whether this protein has function. And the function of this protein is cancer prevention. So if you have the protein, then it helps you prevent cancer, so it helps prevent the overgrowth of cells in the bone marrow. If you don't have this protein, then it doesn't help protect against cancer prevention. So it doesn't help to protect against the cancer, and now you start to see overgrowth of cells in the bone marrow. So this is likely the gene that's actually causing the cancer, but it's all stemming from a mistake in alternative splicing due to a mutation in the splicing factor. So if we have the spliceosome and there are mistakes in the spliceosome that are leading to incorrect splicing, because this research is so recent, there's not too much development for therapy yet, but what scientists are trying to do now is really block this step. So if we know that a mutated spliceosome is causing incorrect splicing, can we prevent that incorrect splicing from being made in order to prevent the cancer from happening? All right, so this is the end of all of what I presented, and I'm just going to summarize what we talked about now. So we learned that splicing is the cutting and pasting of RNA, that alternative splicing creates many distinct RNAs and proteins from a single gene, that mistakes in splicing are common in disease, But scientists are constantly learning more and more about splicing each day to find therapies for splicing-related diseases. Before I take final questions, I want to thank all of you for your attention, and I thank Science in the News for allowing me to give this talk and providing a great platform for science outreach, and all of these other programs at Harvard for making this possible. So happy to take last questions if you have them. Yeah?
Can you describe a little more about, you mentioned splicing factor, and then you mentioned the spliceosome, and I wasn't quite clear on
Which is which.
where and how that fits in that machinery.
Yeah, so the question is, can I describe a difference between splicing factor and spliceosome, and maybe how those are involved in disease? So splicing factor is just a part of the spliceosome. So I mentioned the spliceosome is made up of hundreds of proteins, and the factor is just one of those. So I kind of skipped through what that story was really telling, and I can give a little bit more if that might be what you're asking for. So basically the splicing factor that was mutated appears, the purpose of that factor is to direct alternative splicing, and it binds sequences on the pre-mRNA. And the mutation that happened is it could no longer recognize the sequence that it was supposed to bind to and would prefer a different one with the mutation. So that was what was causing the different alternative splicing events. And that's what was leading to the cancer. And so one important thing that I kind of want to emphasize with splicing factor mutations is that there are some splicing factors that are part of the spliceosome that are really critical for the actual cutting and pasting. Those factors are rarely ever mutated in disease because they're so important that no cell would survive without them, because you really, really need them. The ones that are mutated are the ones that are really dictating alternative splicing and kind of only affecting splicing of a few of, not a few, but a subset of genes. Any other questions? Yeah?
Does splicing affect aging as well?
Question is, does splicing affect aging? I'm sure it does. I don't know, I don't think I have a good example of how it does, or I don't have any studies that can come to mind, but I'm sure there might already be a study that did this, or it would be a cool idea to look over time with aging and see how splicing changes, because I'm sure it does change over time. Yeah?
How many scientists would you say are working on these questions of splicing in the world?
Good question, yeah. So the question is, how many scientists are working on these questions of splicing? I would say a lot. So in terms of the number of scientists that are only studying splicing, and that's kind of their only goal is to understand splicing, I'd say a few hundred. But then there are so many labs that study a disease. So say they study muscular dystrophy and just happen to study a case of splicing with a gene that they're interested in.
How can we keep up with all those splices?
Yeah, there's so many, right? So I don't think I could put a number on it, but it's definitely a lot.
Oh, it was great.
Yeah. All right, thank you guys. Yeah.